Don't tease me like that.
Don't tease me like
I'm
that.
making confused.
No, Amber's here, I know it's micro.
Yep, because Amber is checked on with that class.
Does anyone need a handout for chapter 6? Because I have a couple extra. You need a handout for chapter 6? I've got extra.
Oh, well.
I'm ready for May 6th. I'm ready.
It's just.
Not no offense to you, but
Ready for LASIK?
no, May 6th.
May 6th. Okay, if I said ready for LASIK, I'm
No,
like, what's
I
going
am.
on there?
Can you not see the board?
Not a judgment, but are they gonna put the PowerPoints on the blackboard?
Probably not, which is why I'm giving you guys handouts.
That's fine.
Because that was Clarksville's doing. They set the whole micro thing
Can I
up.
go
and-
yellow?
It's always Clarksville.
Well,
I can go
it
yellow.
usually is.
I can go
Well,
yellow.
in all fairness, I've tried to do it, and they say the files are too big. Like
Okay.
Blackboard Ultra's like, these files have too many gigabytes. We can't transfer. I'm like, you know.
That's all right, I have a second pair of
So
glasses at home.
that's fine. There you go.
If I can find it,
That's
thank you.
fine.
I was hopeful.
Well, that's why. So,
As I
okay.
said, not judgment. Not a judgment.
Let's review. If you're growing microorganisms in the lab, what are some things you need to take care of?
Oxygen, pH,
Uh-huh.
Temperature.
nutrition,
Yeah, they need nutrients to live.
right temperature,
Yep, they need the right amount of oxygen.
Correct osmolarity.
Yep. So they need the right temperature, pH, and osmolarity. Usually meaning they need an isotonic environment. So those are kind of the big 5 things we mentioned last time. If you're trying to grow microorganisms in the lab, you want them to live because you're trying to make an antibiotic or whatever you're doing, you got to make sure they have nutrients. You got to make sure they get the right amount of oxygen, that they're stored at the right temperature, the right pH, and the right osmolarity. Now the nutrients, pH, osmolarity, that's usually from the soy agar. The soy agar petri dish, you know, that's what that agar is basically. It's got nutrients, it's at the right pH, it's at the right osmolarity. What do you have to do for the right temperature?
Incubator.
You have to put it in the right incubator. That means you got to put it basically in the 37 degrees Celsius or the 30 degrees Celsius incubator. Right amount of oxygen, why is that important?
we have some that like need oxygen, we have some that will be killed by oxygen.
Exactly. So you've got aerobic organisms that need oxygen, you got anaerobic organisms that can be killed by oxygen, and you have some that can go both ways, like the facultative anaerobes. What I do basically when we're making those cultures, honestly has a lot to do with, you know, either a loose cap or a tight cap. And what do I mean by that? Well, if you've noticed when we've done like the cultures to look at gram stains and all that, I've got them in a little test tube with a little cap on it. It actually says in the instructions this is a loose cap microorganism, meaning when you put it in the incubator, you got to loosen that cap up so they get oxygen. If it's something like the Bacillus that doesn't like oxygen, it'll say this is a tight cap organism in the instructions. You have to tighten that cap. So practically speaking, that's what you do in the lab to make sure they're getting oxygen or not getting oxygen, depending on what they are. So those are the 5 big things we talked about last time. Did you find it? All right. So where do we get cultures in the real world? Yes, usually from blood, urine, a wound culture, sputum, which means bronchial secretions. So in the real world, if you got somebody who's sick and has a fever and you're suspecting an infection, this is typically where you get the cultures to test, either from their blood, their urine, their skin, if they've got an open wound from diseased tissue, and or their lungs, bronchial secretions, if you suspect they have a respiratory infection. Now, how to obtain a pure culture? First, let me tell you what a pure culture is. A pure culture is a colony of bacteria cells that grow from one dominant microorganism. I'll say that again. A pure culture is a colony that originates from one dominant bacteria cell. Well, how do you get a pure culture? Well, you have to do what's called an isolation technique. And the most common method is what's called streak plating. So what you do with streak plating is, let's say you have a sick patient and you take a blood culture and you take a sample and then you streak a sample from that culture on the petri dish. Well, here's what you do next. You sterilize your inoculation loop. You don't go back to the original source. You go to the first streak and take a sample from that and make a second streak. And then the next step is you sterilize the inoculation loop. You don't go back to the source. You go to the second streak and make a third one. And sometimes you even make a fourth streak. What's happening each time you do that? You're basically diluting and isolating the microorganisms so that by the time you get to number 3 or really number 4, you've isolated the dominant microorganism. You basically isolated the most prevalent microorganism that's likely causing your patient to be sick. So that's known as streak plating. You're making multiple streaks, but they're coming from one another. They're not coming from the original source. You only use the original source for the first one. Then you take a sample from 1, make a second streak, take a sample from 2, make a third. You can take a sample from 3 and make a fourth. Notice there's a whole bunch of bacteria here, so much you can't even distinguish individual colonies. Same with number 2. Once you get to number 3, you can start seeing individual colonies. And by the time you get to number 4, it's just individual colonies. And if you tested, these would all be the same dominant microorganism. So that's called obtaining a pure culture. If you suspect your patient has multiple microorganisms, that's how you would find out which one is dominant. So know the definition of pure culture, and know the most common way to do that is to do what's called streak plating. It's a fairly common lab technique. Okay. Now, as far as cultural media basically means a growth medium. So you guys tell me, what do you use in lab?
Soy agar.
We use soy agar. So what exactly is soy agar? Well, agar comes from algae. It's a nutrient broth, usually liquid. So in this case, it's soybean broth. Soybean broth's like chicken broth, but made of soybeans. It's got lots of glucose and amino acids and all the nutrients that bacteria would need. But you don't want to keep it in a liquid state or it tends to go bad. So what do they do to make a soy agar petri dish? They add the agar. Agar is a carbohydrate or sugar from seaweed. Once you mix the agar with the nutrient broth, it makes a gel. And that's what makes that soy agar gel at the bottom of the petri dish. Because that gel can last for a year or 2 if you put it in the refrigerator. So that's basically how you make the medium or the growth plate, where you're going to grow your microorganism. You can also do agar in a test tube. We did this when we did the sterile technique lab, if you remember that. And this is called a slant. So if you use a slant tube, that's a test tube that's got agar inside it. Now there's multiple different types of media you can do. Soy agar is just one of many. Soy agar is the most common because pretty much every bacteria can grow on soy agar, with maybe one or two exceptions. And it's got glucose, it's got all these nutrients, calcium, magnesium, the amino acids, etc. But different organisms will look different. So on the left, this agar plate has a pH of about 7.3. And what do you see when you look on that? Yeah, lots of bacterial colonies. You can tell they're bacterial because they're very well-defined circular colonies of different colors. So probably different microorganisms. And when you see a colony, remember, you're not looking at one individual bacteria. You're looking at probably a few 100,000 bacteria kind of clumped together to see a colony. What do you see over here on the right? These are fungi. Remember when I said fungus spores usually look fuzzy-wuzzy? Well, there you go. And notice what pH is ideal for growing fungus. 5.6, which is acidic or basic?
Acidic.
That's acidic. So remember I mentioned the other day that fungi grow best in acidic environments? This is why if you're trying to isolate a fungus, let's say you want to grow bread mold to make penicillin, you're actually going to use a different type of agar that's actually more acidic than soy agar. And the fungi will actually grow much better. So once again, kind of manipulating the medium, if you will, to grow the organism you're trying to grow. All right, this is one of my favorites. And pay attention to this one because this is pretty much what you're doing in lab next week. This is the blood agar / strep throat test. Next week in lab, you guys are going to do strep throat cultures on each other. Has anyone ever done a strep throat culture on somebody before? Yeah, what you do, you have to take the Q-tip and you jam it in there as hard as you can. No, I'm joking. I'm teasing. You very gently go into the throat and take a sample. And usually you streak it on blood agar. Well, you might be asking, well, what is blood agar? Blood agar is what we call a differential medium. Very important. It's a differential medium. What do I mean when I say that? A differential medium is a special type of growth plate that's used to distinguish between different microorganisms. So that's where the word differential comes from, is different microorganisms will grow different colored colonies on this particular growth medium. So what blood agar is, it's basically soy agar plus 5% sheep's blood. So they take regular soy broth, they add 5% sheep's blood to it, and then they add the agar to make it into a gel. And it literally looks like blood. Well, why do you use blood agar? This is typically what historically used to diagnose strep throat. I'll explain why. If you were to streak strep onto a blood agar plate, specifically strep pyrogenes. These guys know what strep pyrogenes is? You should.
It's if it gets in your blood, causes scarlet fever.
Uh-huh. Yeah, it's a very pathogenic form of strep. It gives you strep throat. If it gets in your bloodstream, it can give you scarlet fever, which is no fun. If it gets in your flesh, it actually gives you necrotizing fasciitis. What's that? That's the flesh-eating bacteria. The reason it does that is, what did I mention about gram-positive bacteria? What do they like to release in the environment? Yeah, they like to release exotoxins. So gram-positive bacteria, usually in the strep or the staph family, they love to release exotoxins into the environment. Exotoxins are usually digestive enzymes, and they love to break down proteins. Why do they want to break those proteins down? So they can absorb them. Yeah, remember from the previous chapter, most cells can't absorb a great big protein, so they break them up with those digestive enzymes. Now they suck up all the amino acids like a milkshake, basically. So that's why they're basically so corrosive when you put them in cells. So strep pyrogenes has such a strong exotoxin, if you put it on a blood agar plate, you get what's called beta hemolysis. You basically get a golden yellow colony. And why is it golden yellow? Well, because it's destroyed 100% of red blood cells it came in contact with. And basically, when all those red blood cells die, their hemoglobin basically rises to the surface and gives a golden yellow color. So that's what beta hemolysis is you destroyed 100% of the red blood cells you came in contact with.
And you said, how does it give it on color?
When the red blood cells die, their hemoglobin proteins kind of float to the surface and turns it yellow. Yeah. And historically, up until maybe even 10 years ago, this is how most clinics diagnose strep. As they do the little throat culture, they streak it on a blood agar plate, they call you in 48 hours and let you know if you had strep or not, and they based if you grew golden yellow colonies or not. Nowadays they have like a dipstick test that's pretty much instant, but historically this is how you tested folks for strep throat. Now, sometimes you'll do a throat culture and the microorganisms will give like a green or beige colony. And that's called alpha hemolysis. So alpha hemolysis means basically a green, or I would say beige, sometimes I've even seen it silver colony. And the reason it's this color is only about 50% of the red blood cells were destroyed. So while strep pyrogenes kills 100% of red blood cells and gives you this golden yellow colony, some other microorganisms are not as pathogenic as strep, and they only destroy maybe half of the red blood cells, so their colonies grow either green or beige or silver. Some common ones that would do this would be Staph aureus, because a lot of us have Staph aureus in the back of the throat. Or there's some weaker strands of strep, like strep pneumonia or strep viridans. These three are actually quite common in the human throat, so they'd actually be considered normal microbiota, which means normal microorganisms in the human throat. And for a young, healthy person with a good immune system, they're usually not pathogenic. Now, if you got like an 80-year-old with a compromised immune system, then they could potentially become pathogenic. But for the majority of the population, they're not. So these guys will give you alpha hemolysis. Now, there's a third type called gamma hemolysis, which is basically negative or no hemolysis. This means no red blood cells were destroyed. Well, the problem with this, I always have a little bit of a problem with this name. Do you see any colonies of bacteria on there?
Yeah.
No. This is just the indentation from the inoculation loop. Yeah, you see 0 growth. Because these microorganisms don't kill red blood cells. These would be your gram-negatives. Because remember, do gram-negatives release exotoxins? No, they got endotoxins. So since gram-negatives don't secrete exotoxins, they don't destroy red blood cells. So if you took E. coli and streaked it on a blood agar plate, all you would see is where you marked it with the inoculation loop. You would actually see no bacterial growth. So this is why blood agar is kind of the gold standard for what's called a differential medium. A differential medium is used to distinguish between different microorganisms. So another way of putting that is that different microorganisms will grow different colored colonies on this growth medium. And that's how you can tell if your patient has a strep infection, or if they have a staph infection, or a minor strep, like strep pneumonia, or if it's a gram-negative. So gram-negative, nothing will grow on blood agar. So you're basically distinguishing between different microorganisms. And there's a whole list of differential mediums. But by far and away, the most common one is blood agar. And that's what we're going to use in lab next week. You guys are going to do throw cultures on each other. It'll be fun. So just be gentle when you do them. Don't make your partner throw up. Or try not to. Any questions on blood agar? Does this make sense? Yeah, you definitely want to take a picture of this before I erase it. So I'll just tell you now, this will be on your lecture test and this will be on your lab test. It will be on both. That's how important it is. So teasing, but go ahead. Take a picture. Won't photobomb it. Yeah, I will pause for a minute because this is so important. I want everyone to get a picture of it. Like I said, this will be on your lecture test, this will be on your lab test, this will be on your final exam. So, if you know it now, you will thank me in a few weeks. Does this make sense? And we're going to do a whole lab based on this next week, so that will kind of drive the point home. All right, everyone got a picture?
Yes.
Okay, cool. What else looks important? We're going to skip some of this because some of this is just beyond the scope of this class. But this is important. So how do most microorganisms reproduce?
Binary fission.
Binary fission. So probably 85 to 90% of your bacteria reproduce by binary fission. You guys tell me, what is binary fission?
Watered down mitosis.
Yeah, honestly, that is accurate. It's basically a watered down form of mitosis. Basically, there's 4 steps. Number 1 is the bacteria makes a copy of its DNA. And remember, its DNA is just in a single chromosome called a nucleoid. So it literally just makes a copy of its nucleoid. And then the cell actually stretches a little bit. After it stretches, then it starts to invaginate the cell wall in the middle. So you get what's called a septum. You can kind of see where the middle of the cell is starting to pinch together. And then finally it will form what's called a cross wall. and then it'll pop apart into 2 identical daughter cells. So a much more watered down version of mitosis, if you will. How fast can your average bacteria go through binary fission?
30 minutes.
30 minutes or less, just like Domino's Pizza, 30 minutes or less. So this is why you can grow a whole colony of thousands of bacteria within 48 hours. Because if you give them enough nutrients, which you do with the soy agar, in the right temperature, they're just going to eat and reproduce, and eat and reproduce, and eat and reproduce. Each one of them can make a copy of itself within a half an hour. So within 48 hours, you can have a whole colony of thousands of bacteria. You couldn't grow like a whole bunch of puppies or kittens in 48 hours. I wish you could. That'd be awesome. But,
Puppy lab.
yeah. But you can with microorganisms. And this is why. Because their reproduction is so simple and quick. This is why bacteria grows so quickly, if you will, if you give it the right conditions. All right, so definitely no binary fission. Now, this is something else you need to know. It's called the typical population growth curve. This is basically the life cycle of bacteria in a petri dish. Let me ask a question. If you streak a petri dish with bacteria, put it in an incubator, does the bacteria colonies, do they live forever? How long do you think they last?
3 or 4 days.
They last about a week, give or take, 5 to 7 days. And why do they only last maybe 5 to 7 days? Yeah, you run out of nutrients. You've only got like a finite amount of nutrients in that soy agar. Eventually, they're going to run out. So typically, the lifespan of bacteria in a petri dish is about a week. That's why, if you notice with our labs, I try to set it up where we streak stuff on Tuesday and then look at it Thursday. Because if we come back like next Tuesday and look at it, probably half your colonies are going to be dead. And you wouldn't get as good results. So, see, everything's by design. So here's the different stages of the population growth curve. For the first 12 to 24 hours, you get what's called the lag phase. Basically, if you were to look at the petri dish, like let's say you had a little camera inside the incubator and you're looking at the petri dish, you would see nothing. Because at this point, the microorganisms are kind of scoping out the environment, they're kind of hunting, finding the nutrients, all that. Now, between 24 and 72 hours, that's where you would see lots of growth. And this growth stage is called the log or exponential phase.
You said how many hours?
I would say between 12 and 72.
Okay, sorry.
Yeah. The reason it's called the log phase, it stands for logarithmic or exponential growth. Basically, I won't make this into a math equation, because binary fission happens so quickly, they grow, we say, logarithmically or exponentially. They grow much faster than most organisms. So that's why it's called logarithmic growth. I'd have to get Jack Smith in here to explain the actual math behind it. But they grow to like the umpteenth power, and that's why they grow so quickly. So this is the log phase. So between 12 and 72 hours, the bacteria have found the nutrients, they're eating and reproducing. They're eating and reproducing. So they're eating, going through binary fission, eating, going through binary fission. Everyone's happy. And this is where you'd see the most colony growth. And that's why we typically look at the Petri dish after about 48 hours or so. Now, after about 72 hours, you'll notice something interesting. This is called the stationary phase. It means your colonies aren't getting any smaller, but they're also not getting any bigger. They've kind of plateaued, if you will. Why have things plateaued in the stationary phase? Not all of them. They've used up 50% of the nutrients. So by the time they get to the stationary phase, half the nutrients are gone. And that's why it's stationary, meaning half the cells are eating and reproducing, half are starving to death and dying. So for every cell you have that eats and goes through binary fission, you have another cell that starves and dies. So the population is at a steady state. It plateaus. The colonies aren't getting any bigger, but they're also not getting any smaller. But by the time you get to about day 5 or 6, you start what's known as the death phase. I think that's a little morbid, but that's what they call it, the death phase. And what's that basically mean?
You're dying.
Nutrients are gone. So since nutrients are essentially gone after 5 to 6 days, you have a lot more folks kind of starving and dying than reproducing going through binary fission. So what you'll look, if you have a petri dish in there like up to a week, the colonies get smaller, smaller, and eventually just wither away. By day 7 or day 8, it's like they're pretty much gone. So this is your typical population growth curve for bacteria on a petri dish. So know these phases. So if you were going to experiment with this population, like once again, let's say you're trying to make a new antibiotic from it, what 2 phases do you think you would probably utilize most? The log and the stationary. Because the log phase is where it grows, where you get the most growth. And stationary, you still have enough of the population still alive. Lag phase, they haven't started growing yet because they're still trying to figure out where the nutrients are. And death phase, everyone's dying off because they've run out of nutrients. So the log phase and the stationary phase is where you would do the most work. And to be honest, you get about 3 or 4 days where you can actually use the culture. So, does that make sense? Good. So, know this. There you go. And with that, guys, I think that is, that's the end of chapter 6. Yeah, moving right along. So, any questions? No? Clear as mud? All right. Now we're going to mosey on to chapter 7. We will not finish chapter 7 today. I'll just already tell you that. It's actually an interesting chapter, but it's a little dense. It's on genetics. And, don't. No whining. I printed you out a big, beautiful handout for this. You're welcome. So this should make your life easier. So there you go.
Big, beautiful handout.
Yes, big, beautiful handout. I could have so much fun with that. It's the best handout you've ever seen. It is beautiful.
Never been a handout like this one.
Best handout in the world. No, just kidding.
Greatest handout of all
With
time.
chapter 7, we're going to cover genetics of microorganisms. And it kind of rolls into chapter 8. Chapter 8 is the last chapter before the next test. And chapter 8 is on biotechnology or gene splicing. So chapter 8 is actually really cool. I teach you guys how to do cloning, how to clone animals and stuff. So chapter 8 is fun. You got to understand chapter 7, though, before you can jump into chapter 8, or it won't make any sense. So just bear with me with this chapter. It starts off a little, so-so, but then it gets really interesting towards the end. So a couple things we will look at. So what is genetics when we talk about genetics?
Study of inheritance and inheritable traits as expressed in an organism's genetic material.
You read that perfectly. Now, what's that actually mean?
It's just the study of DNA and traits
Study
that you
of
can
genes.
inherit. Yeah.
Yeah. Who can tell me what's the definition of a gene? It does, but
DNA.
a little more detail.
So
Uh-huh.
you get from both mom and dad?
That's chromosomes, but yeah.
Oh, I was
You're okay. You're in the ballpark. Okay.
Is it the one spot where they join?
Not quite. I'm sorry. Let me give you the definition of the gene. Write this down. I'm just going to tell you, write this down. Because people talk about genes all the time. Most people can't give you the definition of a gene.
It's the way the DNA
No. A gene,
She's still trying.
I appreciate the effort. Okay.
You're
I'll
done.
just be
A
quiet.
gene is, no, I appreciate the effort. You're doing good. Is a specific set of nucleotides
Oh, yeah.
within DNA that code for one specific,
Protein.
anyone know?
Protein.
Protein, yes, right there.
I knew one word.
That's it. A gene is a specific set of nucleotides within DNA that code for one specific protein. So we're going to go over the structure of DNA. But if you remember, in general, DNA is a double-stranded helix. It's made of millions of nucleotides. Because nucleotides are kind of the building block of DNA. So, long story short, what a gene is, it's a specific segment of nucleotides. We'll say this is gene 1 within DNA that codes for one specific protein. That's it. And in your DNA, you've got millions and millions and millions of genes. So they potentially could code for millions of different proteins. Now they don't, because are most of your genes expressed? No. Of your millions and millions of genes, maybe 25 to 40% of them are expressed. That depends on which genetics journal article you want to read. I'll give you an example. My dad's brother was 6'8", and my mom was blonde and blue-eyed. I obviously didn't express any of those damn genes. I'm a 6'8" blonde basketball player right now, probably making millions of dollars, but I'm not. So you can have millions of genes, but probably less than 40% will actually get expressed and become part of your what we call your phenotype. We'll talk more about that. So that's what a gene is. A gene is a specific set of nucleotides within your DNA that code for one specific protein. Know that definition. And let's say this is DNA here. So, okay, this segment of nucleotides codes for, we'll say, keratin protein. Well, that would be one gene. You take this segment of nucleotides here, it's a different gene. This codes for, we'll say, collagen protein. Then you take a third group of nucleotides down here. These guys code for actin protein, which you have in your muscles. So that's basically what a gene is. It's just a specific set of nucleotides within DNA that code for one specific protein. And that's what genetics is. It's the study of the genes. So it's study of genes and how we inherit different genes from our parents and reproduce, etc. And the genome just means all the DNA in your body. So all the DNA that's in the nucleus of your cells, that's your genome. But once again, you don't express most of your genes. You've got millions of genes. You express maybe 25% to 40% at the most. All right, does this make sense? All right. Big concept of the chapter. If you understand this, the rest of the chapter is going to be a lot easier for you. Is everyone with me? Yeah? Okay. So, nucleic acids, which include DNA and RNA, are made up of nucleotides. Nucleotides are the building blocks for both DNA and RNA. So what's a nucleotide? Well, it's a molecule that's got three things. It's got a 5-sided sugar. That's what pentose means. It means a 5-sided sugar. it's got a phosphate group, and it's got one of 4 nitrogen bases. So this is a typical nucleotide. It's got deoxyribose. That's its 5-sided sugar. So if you look at it, it's got 1, 2, 3, 4, 5 parts to it. It's got a phosphate group over here. And then over here, it's got, one of 4 possible nitrogen bases. So that's what makes up a nucleotide. 5-sided sugar, which in the case of DNA would be deoxyribose. A phosphate group on one side and one of 4 nitrogen bases on the other side. Now, what are the 4 nitrogen bases it could potentially have?
Cytosine,
Yeah,
guanine,
there's adenine,
adenosine,
there's thymine, there's guanine, and there's cytosine.
Isn't yours still 1 for RNA?
That would be for
Instead-
RNA.
Instead of thymine?
Yep. But for DNA, it'd be one of those 4. And you can see here, this one's got adenine, this one's got thymine, this one's got guanine, this has got cysteine. So that's what a nucleotide is. Now, when you look at a DNA strand, honestly, what you're looking at millions of nucleotides chemically bonded together. So that's what makes up a DNA strand. But notice our DNA is what? It's double-stranded. So what you have with our DNA is you've got one strand of millions of nucleotides going in one direction, and then you have a second strand that's got millions of nucleotides going into the opposite direction. And the reason they're going in opposite directions is to make sure the nitrogen bases face each other in the middle, because that's honestly what holds the DNA strands together. So we often say DNA is basically made up of what we call a sugar phosphate backbone held together by nitrogen bases in the middle. All right, let me say it again. Each strand is basically a sugar phosphate backbone, and then you have nitrogen bases holding the 2 strands together in the middle. Why do I call this a sugar phosphate backbone? Because with each nucleotide, the strand part is from the sugar and the phosphate. So you can see the deoxyribose sugar here, and then the phosphate here. And then up here, same thing. You got the sugar, and you got the phosphate. So for these millions of nucleotides that are chemically bonded together, the strand portion is made up of these sugar and phosphate components. And same thing with the other strand coming in the opposite direction. You've got the sugar, and you got the phosphate. And you got the sugar, and you got the phosphate. Okay, does that make sense? Now, because these guys are going in opposite directions, their nitrogen bases from the nucleotides are facing each other. And how do these guys bond together? They have a specific format.
Adenine and thymine bond together, and guanine and cysteine bond together.
Yes, so remember with DNA, adenine and thymine always bond together, and guanine and cytosine always bond together. So if you look with this, A always binds with T, G always binds with C. So you'll typically see it abbreviated like this. A and T always bind together, G and C. So adenine and thymine always hydrogen bond together. Guanine and cytosine always hydrogen bond together. And honestly, it's this hydrogen bonding of the nitrogen bases, that's what kind of glues the two DNA strands together. And that's how you get what's called the double-stranded helix. So that's what Doctors Watson and Crick came up with in 1956. They called this the, double-stranded helix. All right, does that make sense? And once again, what you're looking at just millions of nucleotides chemically bonded together. But this strand, they're going in one direction. This strand, they're going the opposite direction. But that allows their nitrogen bases to face each other so they can bond and stick together. If they were both going in the same direction, these nitrogen bases would be way over here, and the two strands would not stick together. Does that make sense? And once again, what's a gene? Well, you take these set of nucleotides and they code for one protein. So this is a gene, they code for one protein. You take these nucleotides over here, this is a different gene that codes for a different protein, etc. So once again, that's the definition of a gene. It's a specific group of nucleotides within DNA that codes for one specific protein.
Is it always 3, like codon? Is it always 3 that?
No, you're thinking of RNA. That's
Okay.
different. No, you could have like 200,000 nucleotides in one gene, and you could have 50,000 nucleotides in another gene. So it's very random.
Okay.
Good question. All right, does this make sense? Yes? Okay, because you understand this, and the rest of the chapter honestly isn't that bad. Okay? Okay. Yeah, go ahead and take a picture. Okay, everyone good? All right. Okay. Now, remember, there is a difference between the genomes of prokaryotes like bacteria and eukaryotes like humans. With prokaryotes, remember, they don't have a nucleus, they just have what's called a nucleoid. So prokaryotes actually are haploid. They've only got one chromosome. And if you remember, that's what a bacteria, basically, their DNA is. So this is a bacteria cell. They've just got one giant chromosome that's usually circular. And it's not a nucleus, it's a nucleoid. And this nucleoid holds all the genes and all the DNA of the bacteria. And it's just kind of floating in the cytoplasm. It just kind of goes back and forth within the cytoplasm of the bacteria cell. So bacteria are always haploid. They've only got one chromosome. Who remembers what a plasmid is? We'll talk more about that later in the chapter. Anyone remember what a plasmid is?
It's the copy of the...
Copy of a gene. So we'll talk more about this when we talk about conjugation later. But basically, if you got a bacteria cell and it mutates, it makes like a resistant gene, because most plasmids are resistant plasmids. So say it's resistant to penicillin or whatever. It actually will make a copy just of that gene, not of its whole nucleoid, but just that gene, into what's called a plasmid. So that's what a plasmid is, just a copy of one gene. And it will send that plasmid to another bacteria in the same colony through a process known as conjugation. We'll talk more about this later in the chapter. So if you remember conjugation, it makes what's called a pilus. That's like basically a little bridge with its cell wall. And then it's going to send that plasmid over to the second bacteria. What's the second bacteria going to do?
Send it on.
Well, it's going to take it into its own nucleoid first. And now this guy is resistant to penicillin. But then, like you said, it's usually going to pay it forward and make its own plasmid and then send it to a third bacteria cell, etc. And that's how basically bacteria cells in the same colony pass genes to one another. So eventually you have a whole colony that's resistant to penicillin or whatever.
So how fast is that conjugation? I mean,
That's a good question. No one really knows. It's pretty quick though.
faster than they reproduce or slower?
I won't say faster than reproduction, but pretty quick. So usually if one guy gets this, or girl, I don't know, they don't really have genders. If one bacteria gets this, then usually the whole colony gets it within a day. So, now most plasmids are resistant plasmids. It makes them resistant to an antibiotic or something harmful environment. Occasionally you get what's called a fertility plasmid. That's a gene that makes them reproduce even faster.
Oh, great.
Like they could reproduce within 15 minutes instead of 30. So that's called a fertility plasmid, and that's the second most common type. So, but we'll talk more about that. We have a whole thing on conjugation later in the chapter. So that's bacteria DNA.
Might look like a video game.
Now, our DNA in eukaryotic organisms is a little more complicated. One, because eukaryotic cells are diploid. What's that mean? Well, we have two sets of chromosomes.
Oh,
Yeah. You get one set for mom and one set for dad. So like for a human cell, AMP2 students, I'm looking at you.
We already took that test.
What's that?
We already took that test. It's gone.
Oh, it's already gone?
We got the
Okay.
final.
You gotta find
How
it.
many chromosomes are in the average human cell?
46.
46. And why does the average human cell have 46 chromosomes?
23 mom, 23 dad.
Yeah, you get 23 from mom and a matching 23 from dad, and that adds up to 46. And that's where the whole 23andMe comes from. Really should be 46andMe, but now that's 23andMe sounded catchier or whatever they made it. So if you're talking about a eukaryotic organism, which could be a person, an animal, a plant, a protozoa, a fungus, whatever, they're typically diploid. They have two sets of chromosomes within each cell. And I'm not going to go through all this again because a lot of this is AMP1 stuff, but for a cell in a eukaryotic organism to make a copy, it has to go through mitosis, which means it actually to unwrap the chromosomes off histone proteins, where you get the individual chromosomes, and then they go through the whole process of mitosis. But I'm not going to go over that again, because I think that's AMP1 stuff. Now, talking about DNA replication in general, and this could be for a prokaryote or a eukaryote, doesn't matter. DNA replication is semi-conservative. What do I mean by that?
One half is used in the new one, and one half is
Mhm.
used in the other one.
Basically, each copy has an original DNA strand and a copy of a DNA strand. So if you have, this is the original DNA here, if you make a copy of it, each daughter DNA, if you will, is going to have one strand from the original DNA and a new strand from a copy. That's what's meant by semi-conservative. And then if you replicate them again, then you get an original strand plus a whole bunch of copies, basically. Okay, let me see. What else looks important? I'm going to spare you from most of this DNA replication because it's extremely boring.
It will put you to sleep faster than Ambien. I'm not joking. So I'm just going to tell you just a couple highlights. With eukaryotic organisms, if you're looking at double-stranded DNA, you have to separate the DNA before you can replicate it. So typically, and I'll be honest, prokaryotic organisms have to separate their DNA, even though if it's single-stranded. So usually you have to have an enzyme that separates the DNA. And that can be DNA gyrase, which we've talked about, or DNA, in this case, helicase. What these guys do is they separate the strands so the strands can be copied. The other big thing I will take away from this is the enzyme that actually copies DNA or makes copies of DNA is called DNA polymerase. So that's Polymerase, DNA polymerase. Write that down. That's the actual enzyme that makes copies of your DNA. So what basically happens when our cells want to make a copy, you got to separate the strands first with, an enzyme like DNA gyrase or DNA helicase. Once you separate those strands, then DNA polymerase comes in and actually makes a copy of each strand. So that's how your DNA makes copies of itself. That is the very short answer. I saved you guys about 6 hours of a very boring lecture I had to sit through in medical school. We were all ready to jump out the window about halfway through. So that's the meat and the potatoes of it.
And you said prokaryotes.
They also use DNA polymerase to reproduce. Theirs is just a little different because it's typically one strand. But they're showing here, you can see DNA polymerase comes in, it goes down the strand, and it's making a copy of it as it's kind of going down. Okay. Now, this is getting more into the heart of the chapter. You guys need to know the difference between the genotype and the phenotype. So who can tell me what's the difference between genotype and phenotype?
Genotypes the ones that you have, phenotypes the ones that you've expressed.
Correct. So genotype is all the genes in your genome. The millions and millions and millions of genes you have in your DNA, that is your genotype. Now, once again, are most of your genes expressed? No. Maybe 25 to 40% of your genes are expressed, if that. The genes that do get expressed become your phenotype. And what's your phenotype? Those are usually your physical characteristics and your behavioral characteristics. So your physical features, if you're tall or you're short, or if you have blonde hair or brown hair, if you have blue eyes or green eyes or whatever, that's your phenotype. That's what genes that were actually expressed and basically made you what you are. But also, they're the genes that get expressed that have the functional traits of the organisms, or if you're talking about people, this is a nice word for personality. Do personality traits run in families?
Yes.
Yeah, I've been a psychiatrist over 20 years, trust me. I treated grandparents, parents, and kids. And it's like, yep, everyone's got the same problem. Everyone's in denial about it. Yeah, it's pretty much the same. What's going on with grandpa is going on with the grandson, or vice versa. So 9 times out of 10. So that's the phenotype. Genotype is just all the genes you got in your DNA. Phenotype are the select genes that actually get expressed. And the phenotype is both your physical features, but also your functional traits, or in humans, personality traits. So what do I mean when I say a gene gets expressed? What does it have to go through to get expressed? It's got to go through two things to be expressed. And we actually taught this in A&P 1. It's got to go through transcription and translation. Yeah, I know. Don't worry, I'm not going to have you guys memorize this again. But this is what we mean when a gene gets expressed. So here's a gene. Here's a set of nucleotides within your DNA. The gene has to get transcribed first into a molecule of messenger RNA. And then that molecule of messenger RNA has to get translated by a ribosome to make a polypeptide, which is a protein. So once again, a gene is coding for a specific protein. So when we say a gene gets expressed, that basically means that gene has to go through transcription, and then it's got to go through translation. And then after that, you get a protein, and that protein contributes to your phenotype. So that's kind of the jump between genotype and phenotype. Genotype is all the millions of genes that you have in your DNA. Phenotype is the percentage of those genes that actually get expressed. And once again, when I say they get expressed, that means they got selected to be transcribed to messenger RNA and then to be translated into a protein. So, and then those proteins make up your phenotype. Does that make sense? Yes? No? Kinda?
Maybe.
Maybe? What do you, what's...
This stuff I'm just not good at. I have to really work hard for it. So I just
Okay.
have to really study a lot.
Okay, that's okay. So just think, genotype is just all the genes you got. Phenotype are the genes that actually get expressed that make up your physical characteristics and your personality characteristics. And to go from genotype to phenotype, gene expression means this gene went through transcription and then translation. Transcription is where you take a segment of DNA and you make a messenger RNA molecule from it. And then translation, you take that messenger RNA molecule, you hook it up with a ribosome, and you make a polypeptide or protein. And that protein contributes to your phenotype. Does that make a little more sense? Okay. And we would back up one slide. This is known as the central dogma of genetics. Write that down. The central dogma of genetics. First of all, does anyone know what dogma means? It's an old term. It's a Latin term from literally like 2,000 years ago. Dogma just means law. I don't know why they don't just say central law of genetics. It didn't sound fancy enough. So it said the central dogma of genetics, central law. Basically, the central dogma or law of genetics is DNA is transcribed into messenger RNA, and then messenger RNA is translated to a protein. That's basically the central dogma of genetics. DNA gets transcribed to a messenger RNA molecule, and that messenger RNA gets translated to form a protein. That's it. And once again, that's how a gene gets expressed. It's got to go through transcription and be made into a messenger RNA molecule, and then it's got to go through translation. That messenger RNA molecule has to hook up with a ribosome and make a protein. That's where you say a gene has been expressed. Question?
Back to phenotypes and genotypes. There are some things that are not expressed until environmental. Would those be considered genomes, or are they phenotypes?
What do you mean not expressed until environmental?
Like, some genes don't come out until you have certain. Never mind. I'm lost. It's okay.
Most of your genes have to be expressed before you're born.
Okay.
Yeah.
So it just
Pretty
might
much
not
what you're
be-
born with is the genes you got.
Might not be
So-
activated, but it's
Yeah, that
still
might be a better way.
pheno
They're not going to
thing.
be activated per se, but they're already expressed. So,
Okay.
Yeah, what you got in the basket when you come out is what you got. You know, it may get
It
may
may
not get
blossom
expressed.
later, but
Okay.
the genes did not change. The only way you could change someone's genes is either through gene splicing, which you have to do usually before birth, or mutation, like radiation exposure, which is usually bad because usually the mutations leads to cancer. So, okay. Let's see where we at. I think I'm gonna go ahead and stop there for today. But here I'll probably like, thank God. But once again.